I am the head of Codebase at Ginkgo Bioworks, and looking forward to telling you a little bit more today about cell development kits. And after the break, we'll also have a number of folks from our CDK team who will go more into the details about how you can actually leverage cell development kits for your particular projects. But before I jump into that, as Ina noted, Ginkgo's platform is designed to help you make your products and projects move faster. And we do that via two core parts of our platform. Our Foundry is basically the software and automation that we apply to engineering biology to be able to do more projects simultaneously, and also to advance those projects more quickly by collecting more data than you'd be able to do if you were doing those projects by hand. And that's very important to help us serve you better. However, I'm responsible for the other half of the platform, which is extremely important in making sure that we're actually getting better at engineering biology over time, which is to say we're collecting biological tools, useful genetic code, reusable parts, talented organisms for producing different products. Collectively, that's what we call our code base, and this is a term borrowed from the software world. Effectively, if you're a new software developer, say you work at Google or Amazon or any of these large software companies, you don't start from scratch when you're developing a new product. You borrow code from libraries and modules developed by other developers to help build a new app more quickly. And we think about biology exactly the same way. You know, if we've engineered an organism to produce protein at a high titer, it's probably going to be good at producing many different proteins. So from our perspective, we want to make sure that that code base is reusable and accessible by our customers. However, you know, as Ginkgo has grown and the type of work that we do has gotten more complex, it's become harder to kind of understand, you know, how exactly our code base can help you. So today, we're really excited to talk about cell development kits as a new way to access our code base and help you get onto our platform faster. Why is this important? I would say that we're in the punch card era of cell programming. So probably like many of you, I did my research in synthetic biology as a graduate student. I spent 5.5 years pipetting small amounts of liquid from tube to tube, and that's basically how I got my work done. You know, when I did my PhD, I think about more than a decade ago now, I designed and synthesized, you know, 6 different genes for my entire PhD project, and that was a lot of work. I did a lot of cloning while I was doing that, and effectively, you know, the majority of my time was spent smashing small pieces of DNA together, not actually doing my thesis research. And this is how it's done in most places today. So let's say that you're a founder, you have a great product idea for synthetic biology. You're going to need scientists to advance your product development. If you're starting today, you may say, okay, to start developing my strain, I'm going to need 4 bench scientists to get together to start building the genetic parts to make a commercial strain to produce my product. 4 scientists is not going to get you to full scale production, but it'll at least get you started. How much do those 4 scientists cost? Well, once you hire the scientists and you put them in a well-stocked lab, you know, people need reagents and space and equipment to work. It's going to cost you about $1 million per year just to keep those 4 scientists busy, right? You're probably going to have to hire 4 scientists to get your company off the ground. However, we'd like to say with CDKs, we think that you should be able to start your company and have those folks work on something more interesting than putting pieces of DNA together. So, you know, from our perspective, we think the answer to how many bench scientists you need to build your commercial strain should really be 0. You should still hire scientists, of course, but those folks should be focused on product development. So, you know, Jason mentioned our partner Motif FoodWorks. This is exactly how we work with them. Their scientists are focused on food science, material science, formulation, and they're able to do that and spend more of their time on things that are productive related to actually making their product, because Ginkgo is handling the strain engineering. So, you know, for CDKs, we'd also like, you know, our customers, all of you, to be able to use the platform in the exact same way. This is not a new idea, and the reason I use the punch card analogy in the beginning is that in the software world, different platforms figured out how to enable developers to use their platforms more efficiently. So in the software world, these are called software development kits, or SDKs. A couple examples here from Apple and Microsoft. But the basic idea is, rather than just give people direct access to your platform and say good luck, what you do is you take reusable code libraries, code base. You take documentation. You take licenses so you can actually use the App Store or platform, and you bundle them all together and help developers use your platform in the right way, right? So the iOS SDK gives you all the guidelines you need to be able to write an app that can be put on the App Store. Same with the Windows SDK. And these were invented as a way of packaging together all the components a developer might need to use a platform correctly and quickly. So at Ginkgo, since we work in cell programming, we're calling these CDKs. They stand for cell development kits. What's in a cell development kit at a high level? Ultimately, you're going to need a cell to get started with the cell development kit. So we're building these cell development kits around particular organisms that we've engineered to be highly productive in certain areas. You're going to need genetic code. So, at Ginkgo, the organisms that we're developing for CDKs are organisms that we've worked with extensively over many projects. So that means we figured out kind of the tips and tricks to get started in these particular organisms. Same thing on biological tools. We have all the tools we need to edit and improve these organisms, because again, these are not brand new organisms for us. These are things that we've been engineering across many projects. And finally, and we feel this is important, we wouldn't be offering cell development kits as a way to access our platform today if we didn't feel like the CDKs that we're offering had commercial validation. In other words, these are organisms that are real workhorses, in the industry, and enable us to develop strains that can be commercialized and scaled up. So again, for us and what we're doing with CDKs, the real opportunity here is that it helps you specify what you need so that Ginkgo can build the organism and you can focus on building your product. Again, going back to this example of Motif, they've hired food scientists and formulation experts, and that allows them to focus on what really matters for scaling their business. So today, I'm going to give you a high-level overview of our first CDKs here. These CDKs, we're focusing on protein production. I know that a number of you, on this call are probably working on products besides proteins. Looking forward to talking to all of you at our breakout sessions, because we'll have some breakout sessions for you as well. We don't have a CDK to offer to you today, but it's likely we can get started on talking to you about how to get you on the platform via other means. The first CDK I'll talk about is our Pichia CDK. So, I mentioned for CDKs, really what you need is to package together best-in-class parts and tools to enable us to get these programs off the ground more quickly. Now, other folks after break will be giving you more detail here, but ultimately, as an example, we had to develop better promoter systems. Now, the standard promoter system in Pichia was not something that we really wanted to use here for a range of reasons, including the need to use methanol induction, for example. So rather than develop a single promoter system, we developed pretty complex libraries enabled by machine learning to develop a whole range of different promoters, with our best promoters in industrial processes showing performance more than 20x better than our reference standard for this type of system. Again, and with options including and not including methanol. Similarly, we have a lot of experience in taking Pichia strains from the lab to commercial scale. So this is an example for work that we did with Motif, and again, this is kind of the prototype for the CDK that we're sharing with you today. Ultimately, we had about a year to work with them to develop a prototype strain for them for producing a particular protein. In this case, we looked at more than 300 unique proteins for this particular project, screened hundreds of strains, then went into a round of strain engineering and pathway optimization to release a prototype strain that they were able to produce test amounts of product for in less than a year. And ultimately, that strain actually exceeded the specifications that we had set for ourselves with Motif for that strain by more than 70%. So ultimately, this experience was one that motivated us to say, okay, how do we take those best practices, what's worked well on projects like this? And again, our aim is not just to build a initial protein production strain, but actually put you on a path to commercial scale production. And how do we systematize that and make that more accessible to developers like yourselves? Again, I mentioned, more than a 300% improvement here over the unmodified strains. The second CDK that I want to introduce to you is one that's based on fungi. And now this is a newer CDK for us. We're calling this our period, as we're really looking to bring this new strain onto our platform. And it's relatively new to us because we brought it on board via the acquisition of a company called Dutch DNA. So Dutch DNA, for folks who aren't familiar with them, is a company based in the Netherlands that became part of Ginkgo Bioworks in July 2021. The folks at now Ginkgo Netherlands are experts in engineering filamentous fungi. So perhaps the best known strains in this space are Aspergillus niger and Trichoderma. For today, the fungal CDK that we're presenting to you is an Aspergillus niger-based strain that, again, if you're familiar with the industry, is a type of strain that's really been proven in industrial manufacturing. The unique thing about the strains that we've developed via our Ginkgo Netherlands division is that not only are these strains highly productive industrial filamentous fungal strains, they also have low viscosity. For folks who haven't worked in industrial biotech before, why does viscosity matter? Well, filamentous fungi are useful, for protein production. They're known in the industry for being able to produce proteins greater than 100 grams per liter. But the downside to them is that they create filaments. That's why they're called filamentous fungi. It turns out that you can modify these strains to reduce the amount of filamentation. And that's useful because in a full-scale industrial bioreactor, you don't want filamentation to actually slow down and gum up the reactor and reduce your oxygen transfer rates. So, our fungal CDK is actually a really useful Aspergillus strain that is able to retain high titer protein production while having more of a yeast-like viscosity, which means we're really excited about the ability to use that to drive up oxygen transfer rates and produce high titer production without some of the downsides of using filamentous fungi. And again, we have experience here exceeding more than 100 grams per liter protein production with this fungal CDK strain. And again, we're really excited to learn from all of you about the different types of products you're trying to produce, but we know that mammalian proteins, bacterial proteins, fungal proteins, there are so many different applications for synthetic biology. And the real challenge is being able to produce enough of that to actually do applications testing for product development. So, again, this fungal CDK is a strain that's been tested with many different protein types, and we're really excited about the results that we're seeing there. Okay, so again, you're going to have a chance to talk with our CDK team after the break to learn a lot more about these systems and how to use the CDK. But just as a high-level overview of the two systems, the Pichia CDK, again, we've developed more than 100,000 different expression designs with our best promoter showing a 20-fold better expression level in fermenters than the industry standard. We've collected data in this strain for more than 10,000 proteins, and for challenging proteins, we've demonstrated more than 20 gram per liter intracellular protein production in this strain. The fungal CDK, again, this is beta. We're getting this on the platform over the last few months. But again, what's exciting about this strain is low viscosity phenotype with high productivity, more than 100 grams per liter for certain proteins. We're onboarding this strain into our Ginkgo's Foundries today, so enabling a lot more scale than was previously available at Dutch DNA to further advance the strain. And again, this is a strain with a proven track record for secreted protein production. Okay, so again, we'll have more of a chance later to talk about how to use the CDK platform, but really what we're offering here is an initial CDK that helps you get started on your journey to developing a full commercial strain. And I think one of the advantages of working with Ginkgo's Foundries is that there are many different paths that you might try to take to get to a commercial strain. Using the Foundry and using our automation, we're able to explore many more of those paths that you'd be able to do with your 4 scientists who would cost you $1 million per year. And ultimately, what we want to do with you is help you grow with Ginkgo, as the event title suggests, to walk you through the steps needed to produce a commercial strain. And ultimately, by getting onto the platform with our initial CDK offering, you'll be able to get direct access to our teams that have a lot of experience in doing this kind of development, and we'll be able to develop further work packages that help you get towards that commercial strain. And again, it's really important that we make this offering accessible. So again, remember that example, 4 scientists working at the bench is going to cost you about $1 million per year. For 1/10 of that cost, for about $100,000, we want to be able to get you on the platform and show that we can use our Foundries and Codebase to develop a commercial prototype strain for you. So the idea is being to set this offering at a price point that allows you to get your company started in a lean way while still having access to the best that Ginkgo has to offer. So with that, I'm going to try to play one more video here to talk about one of our first customers for the CDK platform. You're going to hear from the CEO of Humblebee Bio, which is a company based in New Zealand, which is going to be using the CDK to develop their unique product. So I'll let you hear it from them.
Hello, my name is Ryan Graves. I am the interim CEO of Humblebee Bio. We're a company out of sunny New Zealand. We are on a mission to build novel bio-inspired materials to replace plastics, the petrochemical-based plastics. These microplastics are rapidly accumulating in every part of our environment. Our lead target is from a solitary bee. It wraps its larvae in this nest material to protect it. This nesting material, when analyzed in the lab, beats plastic on a bunch of metrics. We are trying to recreate this using synthetic biology and fermentation, and biomanufacture this at an industrial scale to replace plastics in everyday products such as textiles. We've entered a collaboration with Ginkgo to drive our lead target sequence through the CDK program. We are super excited to access Ginkgo's platform. They're adding diversity to our existing R&D, bringing in new tools and a new chassis. We like the CDK structure being in 3 phases. This de-risks the program for us, enabling decisions at each step to be data-driven. Time to solve these problems, plastics pollutants, climate change, is not on our side. These are existential threats. We don't have 10 years to slowly figure out our molecular biology. We need to bring our products to market as soon as we can. We believe Ginkgo's expertise and high throughput platform will accelerate this R&D, way more shots on goal, giving us a higher probability of success and much, much faster. This acceleration is exactly what Humblebee needs. And we're really pleased to be collaborating with Ginkgo.
So there you have it. Again, we'll have these videos shared on the website as well. But again, great to hear from a customer like Humblebee that is actually going to be using the CDK platform to get started with Ginkgo. And we're really excited to hear from all of you to see about how we can use CDKs and Ginkgo's tools to help you move your program and your company along more quickly. So we're about to go into our breakout sessions. So I think you may be able to do that through the interface now. So Jason, Ina, and I will be around to drop into these sessions as well. So look forward to seeing you all there. All right. Thanks, everyone.